Have you ever wondered when aging stopped being a foggy idea and started acting like a counter you could count? Picture this. 1960s. Leonard Hayflick at the Wistar Institute. Rows of flasks with normal human fetal fibroblasts. Nothing exotic, no special sauce, just careful counting. He tracks how many times those cells split. 30, 40, 45. Around 40 to 60 doublings, they slow, they flatten, they change their look, and then they refuse to divide. Not dead, not thriving, just done. People had assumed normal cells, given food and warmth, would go on forever like HeLa cancer cells. They didn't. Stakes, right there. If healthy cells have a built-in limit, aging isn't just damage piling up. There's a program that says stop. And once you see a program, you can ask what it's made of, who said it, and what happens if you push the buttons. Action as it unfolded. Hayflick keeps receipts. He freezes cells, thaws them years later, and shows their divisions add up across time. He shows this isn't contamination, not a bad batch, not a lab fluke. It's normal biology. Meanwhile, the field is still soaked in the belief that cells are immortal in culture if you treat them well. A belief fueled by Henrietta Lacks's HeLa line, which was actually a tumor, with its safeguards broken. That difference matters. HeLa is like a car with the brakes ripped out. It'll go far and fast, straight into a wall. Resistance comes quick. Senior voices push back. You must poison them. Your technique is off. Hayflick answers with data and a freezer full of proof. The WI38 cell line he helps establish becomes a workhorse for vaccines, including rubella. Quiet irony. The same cells that prove limits also help us cheat infectious deaths, letting more people make it to old age where those limits start to bite. Impact. The immortality vibe in cell biology takes a hit. Labs begin to separate normal from cancer in a serious way. The question shifts from how do we keep cells dividing forever to how do cells decide when to stop and what are they protecting? That pivot births a new language. Senescence, checkpoints, and soon, telomeres. Here's the twist that keeps the tension. Cancer cells blow past the limit. Most of them turn telomerase back on, the enzyme that rebuilds the ends of chromosomes. Others use an odd route called ALT, alternative lengthening of telomeres. Either way, they dodge the brakes. It works, for them. For the host, it's disaster. So the limit looks less like a flaw and more like a safety fence. It keeps most of us from erupting into tumors when cells take too many risks. Zoom into a microstory. Picture Hayflick labeling a flask PDL-47, population doubling level 47, like a diary entry. He comes in after the weekend, checks confluence, adds medium, and waits. The cells look bloated, spread out, like they've settled into old age. They're alive, they're metabolizing, they even talk to their neighbors with secreted factors, but they won't cross the line into S phase. He marks the notebook. No further division. That one line becomes a pivot in how medicine thinks about aging, cancer, and what's even possible. Stakes ratchet again when you realize the human angle. If your tissues are built from cells that can only divide so many times, wound healing slows, organ maintenance gets thin, and the odds of breakdowns climb. But you also dodge a world where a hungry precancerous clone can sprint forever. The limit is a compromise. It's not your enemy, it's your bouncer. What nobody knew at the time was the exact counter. Why 40 to 60? Why not 10 or 100? To understand why cells keep score at all, we've got to look at the ends of your chromosomes. Tiny repeating sequences that act like the plastic caps on shoelaces. Every copy shaves them down. When they get short enough, alarms ring. That's where we're headed. Meanwhile, think about the cultural shift. Before this, aging was hand-waved as wear and tear. After this, people started asking, where's the clock? The answer didn't just live in chemistry, it lived in strategy. Your body's long war against rogue growth. The Hayflick Limit said the quiet part. To survive as a multicellular creature, you budget division. You ration youth. You retire cells before they turn. There's one more human beat to hold on to. The same WI38 cells that aged out of division helped make vaccines that saved millions of kids. Those kids grew up and met the lifespan ceiling more often. So the discovery that sets a limit also helps more people reach it. That paradox runs through this story like a seam. Every gain opens a new bottleneck. Every fix reveals the next weakest link. And here's where it gets crazy. If you can find the fuse, maybe you can splice it. If you splice it, maybe you add time. But as cancer showed us, splice too much and you light a different fire. So, to understand the counter Hayflick felt in his hands, we need to go to the tips of the chromosomes themselves, the fraying fuse that decides when the bomb doesn't go off. Telomeres, the fraying fuse at each chromosome tip. To understand the counter Hayflick felt, picture the end of every chromosome wearing a little cap. It's the same 6 letters repeated over and over. TTEG, stacked like beads. That cap is a telomere, and it's not there for decoration. It keeps the real genes from unraveling or sticking to each other. Now here's the catch. DNA polymerase, the copy machine, has a design flaw. It can't finish the last bit on the lagging strand. That end replication problem means each round of division shaves the tip. It's like cutting your hair with clippers that can't reach the last millimeter. You lose a little more every time. Stakes first, because they're real. Every time a skin cell repairs a nick, every time your gut lining renews, you spend a bit of telomere. When those repeats get short enough, the cell reads it as danger. Exposed chromosome ends look like broken DNA. Checkpoints flip. P53 and P21. Whisper. Stand down. The cell enters senescence or bows out with apoptosis. That's the fuse. Too short, and the system would rather stop than risk copying damaged code that could seed a tumor. It's protective, but it means tissues with high turnover run into maintenance limits with age. Wounds close slower. Organs recover less. That's the bill. Action as it unfolded in labs. Through the 1980s, Elizabeth Blackburn maps telomeres in a pond critter. Carol Greider finds telomerase, the enzyme that adds TTAG back on, and Jack Sostak helps show how these caps guard chromosome ends. Telomerase is a reverse transcriptase with its own RNA template, like a tiny factory that rebuilds the fuse. Most of your somatic cells keep it low or off. Germ cells, some stem cells, and plot twist. Most cancers flip it on. You can see the logic. Reproductive lines need immortality. Tumors steal it. Now, why should you care beyond textbook trivia? Because telomere length is one of the few cellular clocks you can actually read. In blood cells, shorter telomeres track with higher risk of heart disease, some cancers, and earlier mortality. Chronic stress, smoking, high inflammation. These don't just feel bad, they seem to tug telomeres down faster. It's not a one-to-one you lose 50 base pairs when you skip sleep kind of thing. And association isn't destiny, but as a signal of wear, it's useful. Here's a human beat. Imagine Greider on Christmas Day 1984, seeing a faint activity band on a gel, evidence of telomerase. That tiny smear would win a Nobel decades later and open a can of snakes. Because the moment you have a way to rebuild the fuse, you're tempted to rewind the clock. People tried it in mice. AAV viruses carrying TERT, the telomerase gene, into aged animals led to better heart function, improved muscle, sometimes longer median life. But even in mice, researchers watch like hawks for tumors, because that same enzyme is active in about 90% of human cancers. Rebuilding the fuse in the wrong cells isn't rejuvenation. It's fuel on a spark. Plot twist. The same liver that makes an old mouse's heart look fresher can hand a dormant precancer a VIP pass. So you get hope with a hazard sign, not a fountain of youth. Complication time. Not all tumors use telomerase. Some use alternative lengthening of telomeres. Basically a recombination trick to copy and paste telomere sequences from one chromosome to another. It's messier, often seen in certain sarcomas and brain tumors. Either way, the path around the fuse is a cancer path. That's the trade written into the hardware. Meanwhile, lifestyle shows up as a quiet nudge on the shortening rate. People with lower systemic inflammation, steady glucose, good sleep, and strong social ties often show longer leukocyte telomeres for their age. Caregivers under heavy, chronic stress sometimes show shorter ones. Athletes aren't immortal, but higher cardiorespiratory fitness trends in the right direction. Again, signals, not guarantees. But if you're choosing levers, these matter more than a shiny supplement stack. Imagine swapping a cart full of powders for 8 hours of sleep and a barbell. Boring, yes. Viral for your biology, also yes. Impact on the big picture. Telomeres turned aging from a vague blur into a checkpoint with wiring diagrams. They gave us a handle to measure change, and a reason why the Hayflick Limit lands near 40 to 60 doublings. They also warned us that trying to brute force division by juicing telomerase everywhere lines up with cancer's favorite cheat. So our thinking shifts. If telomeres are the fuse, then senescence is what happens when the fuse burns low. A cell that won't die, won't divide, but won't shut up either. One last revealing detail to carry forward. Telomeres aren't the master clock of everything. Some tissues rely heavily on stem cells and keep telomerase humming in a tight, local way. Others, like neurons, aren't dividing much, yet still aged. So telomeres are a big dial, not the whole dashboard. That matters because our next stop is what short telomeres actually trigger. A state that can save your life from cancer in your 30s and make your joints ache in your 70s. Senescence, cells that don't die but won't work. So when telomeres run low or DNA takes a hard hit, the cell has a choice. Roll the dice and divide anyway, or pull the emergency brake. Senescence is that brake. Picture a cell that's alive, metabolizing, answering emails, but it's turned in its key card. It won't divide. It spreads out like it's flattening into the dish, ramps up stress responses, and starts blasting the neighborhood with messages. Helpful ones at first. Then noise. Stakes first because this touches real life. Senescent cells build up with age in fat, joint cartilage, blood vessels, even the brain's support cells. A few here and there are fine. Too many, and they clog the works. They secrete a cocktail called the SAFs, the senescence-associated secretory phenotype. That's a mouthful, but think of it as a leaky speaker system stuck on alert. Cytokines like IL-6 and IL-1, chemokines, growth factors, matrix-eating enzymes like MMPs. Nearby cells hear that, stress out, sometimes turn senescent too. Tissues inflame. Repair gets weird. You feel it as stiffness, slow healing, insulin resistance, frailty. It's not one villain, it's a choir that won't stop singing the same anxious chorus. Action, step by step. Short telomeres signal broken end to the damage sensors. ATM and ATR kinases light up. P53 and its sidekick P21 put the brakes on the cell cycle. The P16 INK4A R-bowel pathway locks the door. Mitochondria cough out a bit more reactive oxygen. That feeds back into the stress loop. Chromatin, the way DNA is packed, rearranges, turning on the SASP playlist. The cell starts pumping out IL-6, IL-8, TGF-SPU, GM-CSF, MMP1, MMP3. Meanwhile, it resists death by upshifting survival proteins like BCL2 and BCL-XL. So now you've got a tough, loud, retired cell that won't leave the building. Here's a human-scale moment. A mouse that's been creeping across the lab floor like it's 80 gets an intervention. It's part of the ink attack model. Basically, scientists gave mice a genetic, self-destructive senescence switch they can flip with a drug. They clear a chunk of these zombie cells. Within weeks, the mouse moves cleaner. Its treadmill time ticks up. Its heart and fat tissue look younger under the scope. Later, researchers tried drug combos like dasatinib plus quercetin. One's a cancer drug, one's a plant flavonoid, to hit senescent cell survival wiring. Old mice jump higher. Their fur looks fuller. Median lifespan nudges up. It's not magic, it's housekeeping. Impact shows up in people too, early and cautious. Small pilots report osteoarthritis pain dipping after. Senolytic cycles. Some patients with pulmonary fibrosis breathe a bit easier. Frail older adults post better walking speed. Diabetic kidney disease shows hints of improvement. None of this is a cure, and the trials are still sorting dose, timing, and who benefits. But it suggests that clearing a noisy minority can let the majority of cells work better. It's like pulling cars off a jammed lane. Traffic flows, even if the road didn't get wider. But, and this but matters, senescence isn't a mistake evolution forgot to delete. It's a feature with a price tag. Those same P16's P21 brakes stop tumors in their tracks. A wound needs a burst of SAFs to recruit immune cells and kick off repair, and then it needs that burst to fade. In embryos, a senescence-like program helps shape tissues. So if you carpet bomb senescent cells, you might blunt cancer risk in one place, but trip it in another. You might fix a knee and mess up how a cut closes. Timing and specificity are everything. Meanwhile, the immune system is supposed to patrol for these cells. With age, that patrol gets sloppy. Immunosenescence on top of cellular senescence. Fewer natural killer cells show up. Macrophages chill when they should clean. So senescent cells linger longer, seeding more saps, feeding chronic inflammation, the inflammaging you hear about. It's a loop. Break it at the right spot, you help. Break it at the wrong one, you invite trouble. Get this? Cancer again sits on the other side of the teeter-totter. If you loosen the brakes everywhere to feel younger, precancerous clones notice. If you tighten them hard, tissues that need turnover, gut, skin, blood, start to wear thin. The body splits the difference. Accept some senescence, keep a lid on clones, live long enough to raise kids and maybe grandkids. After that, the math gets fuzzy for evolution. That's why these trade-offs stick. So senescence is the price of safe living in a multicellular crowd. It protects you from a runaway few by retiring the many a little early. And if you can clear the worst offenders, carefully, you can buy better function without yanking out the fire alarms. The question is, why nature picked that bargain in the first place, and what else it gave up to keep us safe long enough to pass the baton. The evolution bet. Die so your kids don't. Here's the part that sounds harsh, but explains almost everything. Evolution doesn't care how you feel at 80. It cares whether you left kids who can leave kids. So it spends big on what helps you survive and reproduce early, and it cuts corners on what happens after. That's the quiet bet under every limit we've seen. Telomeres, senescence, the brakes that hold back cancer but also slow repair. Let's dive into antagonistic pleiotropy, which is a fancy way to say one gene wears two faces. Early on, it's your hype man. Later, it's a tax bill. Think growth and sex hormones. They push muscle, bone, fertility, and fast, wound healing when you need to compete, mate, and raise babies. Same signals also nudge cell division and metabolism in ways that, decades later, hike your cancer risk and strain your heart. From evolution's seat, that's fine. You already passed the baton. The late downside is invisible to selection because selection fades with age. Past your peak reproductive years, the spotlight moves on. Now add the disposable soma theory. Thomas Kirkwood's "you are a budget" idea. Your body is juggling calories and micronutrients and time between three buckets: grow, reproduce, maintain. Maintenance is boring, but pricey. DNA repair, protein quality control, immune policing, all the quiet stuff that keeps cells tidy. In safe environments with low external danger, it's worth investing more in upkeep. You might live to use it. In risky environments, predators, infections, accidents, it's smarter, genetically speaking, to pour fuel into early reproduction. Why fix the roof if the house might burn down this season? So lineages evolve a maintenance spend that fits their world. We got enough to clear childhood, hit puberty, raise offspring, maybe help with grandkids. Not enough to keep everything pristine into year 150. You can see these trade-offs in nature, like signposts. Pacific salmon are the brutal version. Sprint upstream, spawn, crash. Their bodies go all in on reproduction, catabolizing muscle and organs to feed that final push. Not subtle, but honest. Maximum reproduction, zero maintenance. Opossums on predator-heavy islands age faster and die younger than their mainland cousins, living with fewer threats. When the outside world kills you early, there's no payoff for high repair budgets. Bats flip the script. They're small, but they fly, which makes them safer from predators. With lower external depth, selection sees late life more clearly. Over time, bats evolve better maintenance, tighter inflammation control, unusual immune tolerance, and live decades. Same with birds. Lower extrinsic mortality buys time, so the genome puts money into the maintenance line item. Meanwhile, big, long-lived mammals like elephants and whales showcase how expensive that shift is. You don't get to live 2 centuries with a giant body unless you solve cancer several ways at once. Bowhead whales carry tweaks in DNA repair networks and cold-responsive proteins that keep genomes cleaner under stress. Elephants stock extra copies of P53, like genes to kill sketchy cells on site. These aren't upgrades you slap on in one mutation. They're whole system rewrites paid for by slow reproduction, long childhoods, and careful ecologies. The price is years between babies and a huge parental investment. The reward is time. Back to us. Humans look like we hacked evolution because we outlived most hazards with culture. Fire, clothes, medicine, social care. But biology can't rewrite itself on the fly just because we invented seatbelts. Our wiring still reflects a world where infections killed kids and a broken ankle could end a life. So our maintenance budget sits where it's at. Good enough to reach and support reproduction, not bulletproof at 110. Telomeres shorten, senescence climbs, mitochondria fray. The clocks tick because they're part of the safety plan that worked when cancer was a bigger short-term threat than arthritis. Here's where it gets tricky. Some late life traits that look bad might be echoes of early life wins, not mistakes. High insulin signaling helps you grow. Dial it high for decades and you get insulin resistance. Strong inflammatory responses clear pathogens when you're young. Lean on that same setting in your 70s and you get chronic inflammation that cooks tissues slowly. Even bone. Dense bones help a young athlete. By later life, that same biology can calcify arteries. The dial positions are compromises. They weren't tuned for modern lifespan with antibiotics and statins and colonoscopies, so why a ceiling around 115 to 120? Not one lever, many. Antagonistic Pleiotropy gives us late life liabilities bundled with early life assets. Disposable soma caps the maintenance spend. Then multicellular rules add more constraints. You need strong cancer policing, which means rationing division. You need flexible immunity, which risks autoinflammation. You need regeneration, which risks miscopying. Push any one dial hard, say telomerase everywhere, and the others scream. The system stays near a safe operating window because drifting far outside, it makes you either cancer-prone or too brittle to heal. Imagine a car designed to finish a tough rally, not circle the track forever. The suspension is strong, but not serviceable mid-race. The engine's tuned for torque, not infinite hours. You can nurse it past the finish with mechanics and luck, but the blueprint wasn't drawn for lap 500. That's us. Culture and medicine are our pit crew. We keep the car running longer than the bare spec, but the parts were chosen for a different race. Could selection move the line if we lived many millennia in cushy safety? Maybe, slowly. But evolution works through reproduction, not personal desire. And as soon as you try to engineer around these bounds in one generation, flip telomerase on, kill all senescent cells, you collide with the reasons those bounds exist. The bet nature made wasn't die young, it was guard the young, then accept where. We're the first species to argue with that bet at scale. And every argument runs into the same fine print. To buy forever, you must out-engineer cancer, tame inflammation, and fund maintenance in every tissue without breaking identity.
Outliers and oddballs, species that bend the curve.